Hi, everyone. I'm Dr. Marie McNeely, and I am thrilled to have you joining me today for episode 834 of People Behind the Science. Today, we are featuring our guest, Dr. Elaine Guevara. This episode is made possible with support from our sponsor, Innovative Research. Listeners, Innovative Research has provided scientists with quality lab products made in Novi, Michigan for more than 20 years. Their portfolio includes human and animal biologicals, assay and ELISA kits, antibodies, and more. Products can be purchased directly from their website by credit card or purchase order at www.innov-research.com. That's www.innov-research.com. They offer small quantities, bulk purchases, and custom orders. From human whole blood to specialized antibodies, Innovative Research has what you need to succeed in the lab. And today, we can't wait for you to get to hear about Elaine's lab, her life, and her career. So, listeners, get ready to meet another one of our remarkable people behind the science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to People Behind the Science. Today, I am excited to be speaking with our guest scientist, Dr. Elaine Guevara. So, Elaine, welcome to the show today. How are you?
Thanks, Marie. It's a pleasure to be here. I'm doing great. How are you?
I'm doing well, Elaine. Thank you for asking. And we are so excited to have you with us today and to learn more about you and the work that you're doing. So, before we get into the details of our interview, let me first tell our listeners a little bit more about you and how you got to where you are today. So listeners, Elaine is a lecturer in evolutionary anthropology at Duke University. She received her bachelor's degree in biological anthropology from the University of California, Santa Cruz, and her master's degree in biological anthropology from Hunter College. During her master's program, Elaine also worked at the American Museum of Natural History as an archives assistant and subsequently a database assistant. She then received her PhD from Yale University, and afterwards Elaine conducted postdoctoral research at the George Washington University. She joined the faculty at Duke in 2021, and she also served as a faculty member at the University of North Carolina, Wilmington. And Elaine, today we're excited to get to know you as a scientist, of course, but also more broadly as a person. So can you tell me and our listeners, what do you like to do when you're not doing science?
So I'm a big bookworm. I read a lot of nonfiction. I really like investigative journalism, history, some social science, but to relax, my big thing is Golden Age mysteries, which I've read starting from grad school, where that was very salutatory for me. It was always a great way to get my mind off of whatever problem was nagging me. So love Agatha Christie, Dorothy Sayers, Patricia Wentworth, and modern authors in that style. So like Anthony Horowitz. Love mysteries that aren't too dark. That's what I like about the Golden Age mysteries. I also really like biking and just getting outdoors. I love the mountains. I love wildlife. I love my Merlin Bird ID app. Also like games and wine and hanging out with friends.
Well, it sounds like you have quite a variety of different hobbies to keep you busy outside of science. I love seeing these other facets of life and personalities of scientists that I talk to. And you mentioned you love reading. I think this is a wonderful activity to encourage our listeners to do more of. And so do you have particular books, whether they're nonfiction favorites or some of these Golden Age mysteries that you would like to recommend for listeners today?
I have a ton of books, but a couple that I've read recently, I guess both are sort of relevant to my research. But one is Why We Die by Venki Ramakrishnan. He is a ribosome biologist, but this is a book about basically the evolution of aging and mortality. I really love this book. There's been sort of a flurry of popular books about the science of aging, and a lot of them are very hand-wavy. And also, it can just be hard to read a book that touches on your own area because you know it so well, and it might be either boring or you're getting caught up in the minutia and things you don't agree with. But I just thought this book was so good. I thought, looking at the table of contents, that I already knew all the things that it was going to cover, and I just was reading it for my class. I thought it might be good for assigning for my class. And I did end up doing that. But I also learned so many things that I thought I knew. I learned more about them or sort of stories behind the science. And I thought it was really responsible and measured and balanced. It wasn't just a hobby horse type academic book, but it was also still totally engrossing. So that was definitely a huge winner for me. And I also, since then, actually, even more recently, finished The Memory Thief, The Secrets Behind How We Remember, a medical mystery by Lauren Aguirre. And I don't want to give too much about it away, but it's a really engaging account of the neuroscience of memory, but it weaves together personal stories of both patients, also scientists. And I think she does an especially good job of capturing the science, but also what it's like to be a scientist and the challenges of trying to study something that is really a mystery, and the compassion for patients that a lot of people working in this have. So it was very human, but it also had really good narrative flow. I don't know if you've read either of those books.
I have not, and I'm excited to add them to my reading list. Listeners, I'm going to put these on the website for you to find there as well. Why We Die and The Memory Thief. These both sound like excellent books, so I'm glad that you recommended them for us today. And you mentioned that they touch on your research area as well. We sort of alluded to the kind of work that you do, but how do you describe your work to someone who is not in your specific field or perhaps isn't in science at all?
That's actually a hard question because it depends a lot on the context and maybe even just my mood or what I've been doing recently. I've never really gotten a good answer to that. So if I don't have the chance to give a long answer, I usually just say that I teach biology because that's just the easiest thing for people to understand. Sometimes if it's more of a real conversation, I'll say that I study lemurs as a way to introduce it, or that I'm a lemurologist because that's true. But I study really the evolution of primates using molecular approaches. And one of the areas I'm really interested in is the evolution of aging. And within that, I'm really interested in brain aging.
Very cool. I feel like even starting with a lemurologist is a way to get people excited about it because these are just really cool and quite cute creatures.
Exactly. Lemurs are a crowd pleaser, that's for sure.
Oh, absolutely. Well, Elaine, I'm excited to get into some of the details of the work that you do throughout our conversation today, but I like to start off with just talking a little bit about motivation. I think it can be challenging to pursue a career in science, so it can be helpful to have these little motivational quotes or sayings or just mantras that keep you going. So do you have a favorite that you can share with us today, whether it's about science specifically or just life in general?
I really like that you said mantras because I think I am sort of a mantra person. So I don't know that I have any particularly great quotes, but I have some sort of proverbs that are easy for me to remember. And I often find repeating to myself to remind myself of them. So I really like perfect is the enemy of done, which has been attributed, I guess, to Voltaire, but, it's one of these proverbs. The more I know, the more I realize how little I know, which is extremely true in science. Nothing worth doing is easy. I have to remind myself of that a lot. And then a really important one is all work and no play makes Jack a dull boy. So these are things that I honestly do repeat to myself to remind myself because it's just a constant struggle to remember these things.
Absolutely. I think having these battle cries ready in your pocket to get you through that tough moment can be very helpful. And I know in science, not only do you have these cool projects you get to work on, but you get to work with some really cool people. And I think there's often these opportunities to work with mentors and role models and just these really big figures in the field who might inspire you to get to where you are today. So when you think about your own career path, Elaine, are there particular people who really rise to the top of your mind as people who inspired you and helped you in your career?
Absolutely. That's one of the things that I really love about science is that it really does involve so much across generational mentoring and sort of apprenticeship. And really, it takes a village to raise a scientist. And I've been so fortunate because I've had so many really great mentors and I've taken so many different things from my different mentors. So I think I have people that inspire me in some ways, and then some who inspire me in others. So I had an amazing academic grandmother, Dame Allison Richard, who is a lemurologist. She studied with Alison Jolly. She was one of the first people from outside Madagascar working in the country studying lemurs. She established her own field site. And from really early on, she took a really community-based, collaborative, locally grounded approach to conservation. And she's just a brilliant person. And she's written, I should have mentioned this in my book recommendations, she's written a book, The Sloth Lemur's Song, that I also highly recommend. That's a combination of the history of conservation in Madagascar, as well as the island's natural history. So that's someone who inspires me, but in a way, I wouldn't say she's exactly a role model because I feel like she's superhuman,
right?
to me. And I also feel that a bit about Susan Alberts, who's one of my colleagues and also a primatologist who works at a long-term field site, the Amboseli Baboon Project in Kenya. And she's just also someone who has been really innovative as a scientist and is just very sharp. And one of her mentees, who was one of my mentors briefly, was Jenny Tung, who is also a primatologist now at Max Planck. Both she and Susan really inspired me in terms of how rigorous you could be in science. They just set the bar very high and stick to that. And like I said, there's actually so many people, it really is hard to choose because people have inspired me in different ways. But I will mention one more person who is one of my committee members, Rich Lawler, who is also a primatologist and was actually Allison Richards' student. He's a professor at the James Madison University in Virginia, and he's also totally brilliant and a dedicated lemurologist. But one of the reasons he's probably in some ways the biggest true role model for me is that I've known him since my first year of my PhD program, and he always represented someone who really valued both rigorous science and being a really top-notch instructor, but also having a well-balanced life outside of science and having interests outside of science and not letting things get to him so much, the rat race of science, I suppose. So it was really important for me to have someone who wasn't just inspiring in terms of their academic chops, but also in terms of their lifestyle that looked like something that I would actually be able to maybe emulate. So I'm very grateful for that.
Definitely. And Elaine, I like how you highlighted these two different kinds of figures, sort of the people who make you feel like you want to push harder and get to those academic heights, but also the people who show you how to get it done, how to do that work-life balance and be a human as well as a scientist. So I think those are critical.
Yeah, and part of that is that I'm closer to him. I think from afar, people often seem so superhuman. And of course, no one is. But it's also so important to see that there's really different ways to be successful as a scientist, especially in the early stages of becoming a scientist and a researcher and being in graduate school. It can really feel like there's one right way. And if you're not cut out for that, you don't have a place.
Definitely. I think that's such an important message. And I'm curious to hear about your own academic journey. So, Elaine, can you take us all the way back to the beginning and share what some of these first memories you have are of getting interested in science and getting interested in primates?
I had a pretty circuitous journey, which turns out to not be so uncommon, but I think it's important to say. I, as you mentioned, studied biological anthropology in undergrad. And for the listener that might be unfamiliar, biological anthropology is kind of a hodgepodge of things that really spans primatology to human biology and human paleontology and the fossil record. And the fossil record, especially historically, has been a huge part of biological anthropology. And so it's a lot of human skeletal anatomy as a result of that, too. And in fact, a lot of working biological anthropologists are actually people who study bones and people who work in forensics, like the show Bones shows. Mm-hmm. And certainly for me, what drew me into it was the fossil record. So I have always been as long as I've known about it, fascinated by the really deep past and the idea that the Earth used to be a really different place with these fantastical creatures roaming it. The dinosaurs in Jurassic Park, that definitely I was a big fan of as a kid. So I liked the idea of extinct creatures. So that's what drew me in. But by the time I was in grad school, it was clear to me that that's not what I wanted to do. For one thing, I'm not actually, it turns out, a very good anatomist, but it's also sort of descriptive. Not that what I do isn't also. But I think especially with paleontology, that felt like so much the luck of the draw, what you're going to find, when you're going to find it. And I was drawn to something that you can create an experimental design and try to follow it through. So that's how I got into molecular biology. But to back up for a minute, before grad school, I took 4.5 years off after undergrad, and I worked all kinds of jobs. I worked at Trader Joe's. I worked as a child care professional. So really not involved in science much at all. But I think that that sort of work outside of science has given me a valuable perspective. And then, as you mentioned, I ended up at the American Museum of Natural History. So I started as a volunteer. And then I got a part-time job in entomology, which is not at all related to what I studied. But I also think that was a really important milestone for me, or stepping stone, because it was really getting immersed in science, not necessarily in an area that I would have chosen, but I guess that sort of highlighted unifying principles, if that makes sense, in the practice of science. And also how interesting things can be that you don't necessarily expect to be interesting, and the way you can answer different questions using different systems. Part of the project I was contributing to was looking at biogeography of insects in response to changes in climate. So very cool. And just being at the American Museum of Natural History was such an amazing experience because there were all kinds of different scientists, and we had weekly seminars, and the exposure was incredible. And in the meantime, I did my master's degree at Hunter College, which is across Central Park from the AMNH. And my boss basically just told me, you might as well do this because Hunter College amazingly has their classes scheduled basically to suit people who are working. So all their classes are in the evening. So I did that. I basically got my master's degree at night, and Hunter College is just a really special place. It's a super springboard. I think sometimes it kind of flies under the radar in New York because you have these prestigious schools like Columbia and NYU. But the CUNY schools, the graduate school and Hunter College, they have world-class faculty and such great connections, but at the same time are much more accessible, and they do these things like accommodating people's work schedules. So I'm so grateful to Hunter and feel like I was lucky to go there. So yeah, that turned out to be really great, especially the time I was there. There was recently formed a consortium between CUNY, AMNH, the museum, and Columbia and NYU and some other schools all around evolutionary primatology. Because it's a small field, it's very specific, it's niche, but across these different schools, you can get a critical mass of people. And this was sort of around a training program, but there would be seminars, and then people would take classes together from all these different schools. So I actually got to take a class in the human fossil record at the AMNH and a class at NYU, and I got to know faculty from other CUNY schools, from Columbia, from Rutgers. So that was incredible. And at Hunter, I had a really great mentor, Mike Stiper. So he was a geneticist, and that's really how I made my transition into what I wanted to do for my career in primate evolutionary genetics. So that was a really cool turning point for me, where I had this topical background in evolution from working at the museum and from undergraduate, but I didn't have really any experience in molecular biology. But Mike was totally cool with that and really there to offer that opportunity to get into that. So that was also so great for me. So yeah, then I went on to do my PhD and got really focused on lemurs and into the evolution of aging. And then I ended up doing a postdoc in evolutionary neuroscience. So that was a pretty big jump, but I've managed to link that loosely into a research program around molecular primate aging and neuroscience.
I think that is remarkable. And I really love your career path story because I think so many people who are interested in the sciences, they feel this time pressure, this need to rush through things. And I think giving yourself some time and space to figure out what you want to do can be tremendously valuable and can prevent you from rushing down the wrong path to begin with. Can you maybe share your thoughts on how that experience of taking 4 years off, reflecting, and then getting back into science maybe changed the trajectory of your career path?
I was just so lucky because I had this friend who connected me with someone at the museum, and then that's how I got my job and then was encouraged. So I feel like I can barely take any credit for going to graduate school. At the time that I graduated from undergrad, I remember one of my professors trying to plant the seed in my head that I should go to graduate school. But I think at that time I just had such bad senioritis. I've been in school like all my life, basically, by the end of college. And so I really needed and wanted a break. But then pretty quickly, I missed learning, I guess, when I was out of college. But it's really hard to go back once you've left, and especially if you don't really have a pathway forward, like you've planned to take a gap year. I really encourage people to take a gap year. But if it's been longer than that, it's really clear to me now that it was not a big deal to go back. My former mentor was so happy to hear from me and write me a letter of recommendation to get into my master's program, even though I hadn't kept in touch with her. So, of course, I had all this anxiety about reaching back out. But especially now that I'm on the faculty end, it's so clear, like I would just be really happy for any of my students to do that. But you get these ideas about, I don't know, I guess deservingness and everything. And I just completely agree about the idea of feeling like it's already too late to pursue certain opportunities. So even when I was studying in undergrad, I took geology classes because those were most relevant to paleontology. And then I sort of realized my senior year, I needed to be taking classes in the bio department. So believe it or not, I never took a single class in the biology department. And it's kind of ironic that I ended up doing evolutionary genetics because UC Santa Cruz was like a hot spot for that, just not within anthropology. So I do kick myself, but I remember looking at the course catalog and finding all these classes I wanted to take, but then they had this whole string of prerequisites, not just in biology, but to take them, you had to take organic chemistry and all these other classes. So I think it's fortunate. I kind of took, sometimes I feel like a side door into basically working in those things, but without following the traditional path. And of course, that's not always possible. And if I were to go back and talk to myself then, I'd say do a post-bac, do an extra semester or an extra year or summer school. It feels interminable right now. Maybe even take a gap year, take a semester off, but then come back to it because you're so young at that point. And I think ultimately a lot of researchers do end up changing their paths over the course of a career. And that's just sort of, I guess, where your curiosity takes you. And of course, the way science unfolds is very unpredictable. So you sort of follow that along with the opportunities you have. But yeah, it's not really ever too late. And it's definitely better to take the time to figure things out. And I think this isn't what you asked exactly, but maybe I'll also throw in that I think sometimes there's this idea that science is about innate ability and you're like a good scientist or not. And you know that by an early age. And that was really not the case for me. I mentioned that I'm bad at anatomy. I mean, I got a C in anatomy and it ruined my GPA. It was very upsetting.
Oh, no.
And I also didn't do great and had like terrible stress the semester that I took osteology, which is basically skeletal anatomy. I just think people's brains process visual information differently, and I'm not good at pattern recognition or something. So every specimen would just look different to me, and I couldn't really bin them, if that makes sense. So trying to recognize these features, if it looked a little bit different, would be really hard for me. Anyway, I guess the point is I now feel totally comfortable being like, I'm not a good anatomist, but it doesn't mean that I'm not good in the lab. So I've been able to do genetics and learn how to do a lot of basic genetics bench work and analyze genetic data. And it's just a totally different skill set. Of course, there's things in common, like you need to be methodical, you need to be pretty meticulous, you need to have good records. And there are definitely principles to data management and all of these things. But that's another reason it's good to find your interest, but also you can have very varying skill sets. It might come up later, I don't know, but I did field work and that's an extremely valuable experience for me, but I'm also not a great field biologist. Sometimes it's just really hard to know until you try things.
Oh, I couldn't agree more, Elaine. I think you brought up so many important points there. And I think there is this pressure in science. You feel like you have to be brilliant at everything. The GPA comment resonated with me as well, where you're like, if you get that one bad grade, you feel like you're not going to be successful in science. And clearly that's not the case. There are so many examples of this, and your career, I think, is one of them, where you are doing amazing work, even though you kind of had this slightly circuitous, maybe non-traditional route to your current position. But I'd love to talk more about what you're doing in your current position. So, Elaine, do you have a project going on right now that you are just the most excited about and want to share with us?
So I'm at a really exciting stage of a project that has been in the works for such a long time, and it's still surreal to me that we're finished basically with data collection. So this was a project I first envisioned when I was a postdoc that really was merging all the various interests that I had developed at that point. So it's looking at molecular brain aging in primates. So it's a comparative molecular biology project in an evolutionary context. And specifically, we're looking at gene expression and epigenetic changes with age in the cortex and cerebellum in humans and 2 other primates. So just the Friday before this last Friday, I dropped off the last batch of samples at the sequencing core. So we will probably have our last batch of data by the end of the summer, and then there'll be a ton of data analysis. But it's kind of amazing to have gotten to this point because it's just been a really long time with collecting the specimens and figuring out all the methods and then actually getting the nucleic acids and things like this. So the idea, what the motivation is for this project, is to get insights into what aspects of the primate brain are conserved in aging and then what is maybe uniquely human. There's an idea that's still really not rigorously validated, but that human brains are uniquely vulnerable to disorders in aging. It's hard to know for sure just because we lack a lot of the ideal data comparators for our closest living relatives, other primates. Commonly used model species in neuroscience and in biomedical research in general, rodents, their brains are pretty different to ours. So when we look at them, we do look pretty unique, but we share a lot more with other primates, but we also tend to live longer. So there's a lot of open questions about that. And this is supposed to kind of help us tease this apart. And maybe ultimately the goal would be to identify effective interventions that can improve well-being. Because obviously cognitive function is so integral to what makes us human and is so crucial in aging for ensuring people's quality of life. So one other thing, this is kind of like a plug for biobanks and collections, because part of the reason we don't study primates that much in biomedical research is because they're long-lived, and there are also a lot of ethical regulations and practical barriers to working with them. It's just hard to get like a good sample size and certainly across the lifespan. But my postdoc supervisor, who was Chet Sherwood, spent years just amassing post-mortem primate brains from everywhere he could. So all the different zoos across the United States that were willing, different researchers, even different sanctuaries. So over a couple decades, he just has this really uniquely valuable collection of primate brain tissue. And so again, this is all tissue that's been collected from animals who died of humane or natural causes unrelated to the research. But it's now serving this really cool purpose. And of course, there are limitations in what you can do with these kinds of samples, but I think they're really important in being complementary to the model organisms or maybe in vitro systems where you can do more direct experimentation to validate things. It's really important to invest in science long term so that studies like this eventually are possible.
Absolutely. And Elaine, you briefly mentioned just some of the challenges with doing research in primates. And I think it's so critical to talk about the challenges in science because doing science is difficult, right? If it was easy, everyone would be doing it and we'd have all the answers. But I think there are just these tough times, these major struggles that you go through. And major failures, I think, that a lot of people have that people don't really get a chance to hear about because they're not published and no one puts them in their presentations. So do you have an example of something that you really struggled with or a major challenge that you had and how you worked through it?
Oh, it's hard to choose because there are so many. But just to stay on the topic of the grant, I mean, I mentioned that this was so long in the works. And part of the reason was because, as you mentioned, it changed institutions. And so that created all this administrative delays associated with being able to fund this project. And then there was also the pandemic that delayed things. But to focus in on a really specific example, part of it was to do with the actual experiments. We were trying to do flow cytometry to do cell sorting. So basically you use this fancy instrument to separate different types of cells in a sample. And that was necessary because the brain is really heterogeneous in the different cells that make it up. I mean, that's generally true for any tissue if you think about it. There's like different types of cells, but the brain, really, you have neurons, but then you have all these diverse glia cells that play really different roles in the brain. And so a lot of times, if you want to look at a particular question, you need to be able to separate out those cells before you collect data from them, right? Because otherwise you're just getting a lot of noise or kind of averaging across cell types that are doing really different things. So it's like basically biologically meaningless. So all we wanted to do was just separate neurons from the non-neuron cells, the glia. And people have done this. It's not innovative. So we were just thinking, okay, this is like step one. But flow cytometry, it turns out, is like notoriously tricky. And so working with the postmortem tissue made it harder. And the fact that these specimens had been ultra frozen. And then trying to do it for really small pieces of tissue because we were trying to look at really specific regions of interest. That made it harder. And this is true for all mammal brains, but especially for primate brains. It's what it turns out people in flow cytometry call high debris, which I think is kind of funny. So basically your brain is mostly lipids. So it's 60% fat by dry weight. And that's the myelin that coats your neuronal axes. And it's really, really important that you have a brain full of lipids. So it's far from debris. But when you're trying to get a bunch of sticky lipids off the cells so you can study them, that's the hard part because you can't sort them otherwise. There's just all this myelin debris in the samples, and optimizing that was really challenging. And every time we thought we optimized it, there would be another flow run with a complete mystery result. And then to add on to that, it turns out that primate brain cells and the debris in them, or I should say like the other biological material, autofluoresces. So you separate the cells in flow cytometry by tagging them with these fluorophores. So these molecules that when they're excited by a laser, they give off light of a certain wavelength. And many biological materials, including cells, autofluoresce, meaning that they naturally give off light when they're hit with a laser of a certain wavelength. But usually that's not a huge issue because it's going to be a lot more subtle than your synthetic fluorophores that you're using in your experiment. But primate brains, they just really glow. And they really go in the middle of the range of wavelengths. So we were just getting such nutty results. It was just extremely frustrating. And we finally got it working. But this is something we thought we were going to take 10 days to do that took over a year.
Oh, wow.
That's just classic, I feel like, for science. But I definitely had a low point. It was last December, about halfway through this troubleshooting process. And one of the things I've learned in science is that it's so important not to take things personally. But I've gotten much better about that. But this is one of the times where I was not really able to do that. I felt like it wasn't working because I was inadequate as a lab scientist, that I was just not able to figure it out or just didn't have the right touch. And then that turned out to not be the case. But yeah, it was a bit of an emotional roller coaster, but all is well now. It's a good reminder, though, that everything is always more complicated and harder than you think, especially in biology. And it's okay. Sometimes things inexplicably don't work, and that's really frustrating. And you can do a little bit of troubleshooting, and sometimes then you're able to sort it out and it's really satisfying, but sometimes you're not. Biological systems are super complex and there's a lot that we don't know. So that makes it hard, but it's just part of the process.
Definitely. I think you're absolutely right there. And I think during these difficult times, which, like you said, can drag on sometimes for months or years of just trying to troubleshoot what seems like it should be a straightforward procedure, can be really frustrating. So what was the breakthrough moment where you got over the hill and we're like, okay, we've made it, we've got it figured out?
I guess I should back up a second and say that we actually never figured all of it out. So we wanted to separate neurons from glia, but we also were really hoping to separate a couple kinds of glial cells, and we had to throw in the towel. So it can be really hard. I'm really stubborn, and I think that can be a good quality. Persistence is good. But I've also, something I've had to learn is to know when to say, okay, for now, we need to leave this. We need to collect the data that we can, even if it's not all the data we want, even if it's not ideal, we're still getting a lot. So that was really what happened. There were a few different breakthroughs and it was sort of iterative. So there wasn't a particular moment where we said, this is perfect, right? Like we kind of got it to be good enough for the neurons. And that was really exciting. And then we just decided to move forward with that. That was sort of the turning point of saying, like, we're going to get this data. And it's a huge amount of data. Once I made that decision, I feel fine. But when you're asking these questions, you want to know everything all at once. That's how I am.
Definitely. And Elaine, I think breaking down some of these problems into, can we at least solve this one piece, can be a really helpful approach. And also to solve some of these difficult problems in science, you need to have the right tools and supplies. And we'd like to take a moment to thank our sponsor, Innovative Research, and to talk about their portfolio of flow cytometry antibodies and matched isotype controls. For scientists developing biological assays, running immunophenotypic analyses, or characterizing and isolating cancer-initiating or stem cells, Innovative Research's deep catalog of flow cytometry antibodies stands ready to support your research and provide you with the accurate and precise results you need when you need them. Visit www.innov-research.com. That's www.innov-research.com. to view the entire line of Innovative Research flow cytometry products. And returning to our conversation with Elaine, I think when you get one of these solutions to a challenging problem, it's really important to take a moment to celebrate that success, because I think there are so many victories in science, even just these little everyday ones that often go uncelebrated. So do you have a favorite success story, whether it was a small win or a really big one, Elaine, that you'd like to tell us about today?
I completely agree with that and want to echo that sentiment, because I think earlier in my career, I didn't always mark occasions of success, because I think in science, it can often feel like the ratio of success to failure is really low. And I think that once you accept that and are familiar with that, it's okay. And in fact, it's all the more reason that you do need to celebrate the successes, because there are always going to be a lot of failures. And I actually I don't like that there's so much failure, but I do like that in a way science sort of normalizes failure. I think especially in a culture where this idea of individual success and almost perfection with social media and everything is maybe not necessarily healthy or serving most people. And like most human endeavors are going to be a mix of failure and success. And you really can't escape that or gloss over that in science. So with the flow cytometry experiment, for example, we had multiple celebrations kind of every time we made progress with that. So usually it's just having a glass of Prosecco or going out to dinner, or this last time after we finished the experiments, getting some cupcakes. But it's so important to mark those occasions.
Absolutely. Well, thank you for sharing some of these successes that you've had along the way in this project. And I think you're absolutely right. Taking those moments to kind of take a step back is really important. And I think one of the other things that you get to do a lot, and we sort of touched on this a little bit in talking about your career path, you've been working and studying at a variety of different institutions. And I think there are just a lot of opportunities to travel in science, including for field work, which you alluded to earlier. But I think most people don't realize these are part of the career as they're starting to get into it. So do you have a favorite place that your science has taken you, Elaine, that you'd like to tell us about today?
I think the obvious answer is, of course, Madagascar. I haven't been to Madagascar since before the pandemic, since I was in graduate school. So it's been a while. And I mentioned that I'm not a great field biologist, but of course, just having the opportunity to go there, both as being in a foreign country and also the wildlife side of it, seeing the lemurs in their natural habitat, it was really valuable to me on so many levels. The obvious ways getting to have these incredible, unique experiences that I wouldn't have if I were just in the lab. But I think so much of fieldwork too, it's not really fun in the moment. So I can talk about a really fun trip I had to Italy to go to a conference and all the amazing food and stuff like that. Still be my favorite travel for conferences. Mm-hmm. But fieldwork is a lot of feeling like an idiot in my experience, both in the culture and with the language, interacting with people, being misunderstood or misgendered or frightening children unintentionally, trying to fit in and failing miserably, and also just realizing how little just basic physical world skills I have, as well as just being so humbled by my lack of knowledge. I mean, I'm the one who's the graduate student getting my PhD in the sciences and studying these animals, but I just still know like a fraction of what the people that live there know. And I think that you can say that, but it's a little different than being there and really feeling that, and that's something that's really important to remember. And again, it's just really humbling. And I think that's almost always good to be humbled. I'm constantly humbled in science, but especially in this particular way. And it makes for great memories, because even if at the time it's like almost kind of devastating, I feel like the first time I went to the field, it's very comical in hindsight. There's a lot of good shared memories with the people you were with as well. I joke that I studied aging ultimately because the elderly geriatric lemurs were the only ones I could keep up with. And that's not really a joke.
Oh, I think that is absolutely amazing. And I can only imagine going from this sort of academic career path where you've primarily been working in the lab to being dropped out in the field in Madagascar. Do you have an example of one of these stories? Or maybe could you share what it was like that first time you went out in the field and had to sort of make it and do science out in the wild?
Yeah, I mean, I was with a group at an established field site, so I wasn't totally off on my own. And it is at Beza Mahafali Special Reserve. It's a community-based conservation site. So the surrounding community is involved in various aspects of either running the site or even the research. So there's long-term field assistants that have worked there for, in some cases, decades, and they just know the field site in and out. They know all the animals. So I was incredibly lucky. And it's also the dry forest. So primates in broad strokes are tropical animals. And I didn't work in a rainforest, so I'm grateful for that. It wasn't maybe quite as beautiful, but you can see, you can hear, it's relatively flat. So in terms of what fieldwork could be like, it was fairly easy. But I had so much trouble even just finding animals. And then when I would find animals, a couple weeks in, the field assistants would be like, oh yeah, there's actually 6 animals. Mm-hmm. Or I'd be really excited that I saw one. It was that kind of thing. I think when you do fieldwork, you realize what you're capable of, which is more than you think, but you also realize what you're not necessarily capable of. And of course, if you spend a long time in the field, you're going to get better and better at individual IDs and stuff like that. But it's also just so good to be out of your comfort zone. Like there were definitely a lot of times where I've been, yes, this is really happening. And I think in our home countries, at least in the industrialized world, we're often trying to exercise a lot of control over our own lives and surroundings. And when you're in a distant land, you don't have that. But that sort of needing to learn to go with the flow, and that it's okay. And that even if things happen that you don't want to happen, like being sick or something like that, you will probably survive.
Well, it sounds like you had an incredible experience. And I think a tremendous opportunity to learn a lot, both in terms of these field skills and just culturally interacting with people from a world that seemed very different from where you were coming from.
Yeah, absolutely.
And I know in science, you get to meet a wide variety of different people from all over the world and share your research through these conferences like we alluded to, but also just in different labs that you work in. And I think the people in science are often portrayed a certain way in the media. And I think in so many cases, that doesn't represent what the people in real life are actually like. So we like to showcase these funny moments, these creative, amazing, multifaceted people that you meet in science. So do you have an example, Elaine, of maybe a funny memory or a quirky lab tradition or something else that you've experienced that really highlights this human side of science that people don't often get to see?
I don't know if I have like one specific memory, but I'm definitely someone who I really enjoy comedy and humor. And I think also that is a really great coping mechanism when you're facing all these failures. And science is pretty funny because, at least in my case, or maybe especially, lemurs are so goofy. And I think that's probably why I love them, because I'm pretty goofy myself. I've definitely made a number of parody videos with lemurs in iMovie for my friends or students. We have goofy custom Slack emojis of lemurs in our project Slack channel. But yeah, I loved Piled Higher and Deeper. I don't know if you know that comic strip, but that was so great when I was in grad school to be able to read that and really see the absurd side of science. Tom Gauld's comics are really good too. He has a lot about science. And Dr. Glockenflecken's YouTube, if people haven't checked that out, if you're a medical professional or scientist, you have to stop right now and check it out. He is just so great at capturing the absurdity. I mean, there's a lot of funny things in science that you can parody. And when I was in grad school, I would write these comedic sketches. I never actually made them, but that was just something I would sort of do to entertain myself, like about the group dynamics in a seminar or trying to network at a conference, because you just find all these awkward situations. Another really good one is the UCSC Department of Neuroscience's parody video. So Get Data, which is a parody of the song Get Lucky, is probably my favorite one. For Dr. Glockenflecken, How to Publish a Manuscript is really good. Also Poster Session. So obviously, I'm someone who really tries to seek out the humor. And something else that I've done that's not really probably funny, but it's goofy. I have, over the last couple of years, made this board game that I use for teaching in my class. It's the human evolution game. So you're basically in the Pleistocene, like moving around the board as a prehistoric human. And the board is a map of the world. So you go in all these different places and there's like good things that can happen. And then your population size increases and there's bad things that can happen and your population size decreases. So you can get attacked by Procoptodon Goliath, which was this giant Pleistocene kangaroo, for example. And it's definitely sort of a silly way to teach about human evolution, but it's all based in somewhat, at least, reality and what we think we know. So I find that that's nice because this is really fun, amazing stuff. And sometimes we get really bogged down in just memorization and things like that and lose the fascination and curiosity that is what draws people in.
Absolutely. I love these examples that you gave. And I think they really highlight the humor and also the creativity in science, because I think you have to get really creative when you're solving these big, challenging problems. And that sort of leaks over into other areas of your life and your work.
Definitely.
Well, I think having this creativity is critical, like we said, for solving the big problems in science, and many of them remain. So, Elaine, if we gave you everything you could dream up to address one of these big problems, whether it's funding, staff, technology, all the time in the world, all the staff, what is the big problem that you would first want to answer?
I love the idea of that, but it's so hard to answer. I mean, I think probably ultimately I would come down on what is the thing that makes humans prone to neurodegenerative conditions, just because I think that's so important and relevant to people's real life, but also interesting from a neuroscience and evolutionary biology perspective. So, of course, I should say, I mean, I don't want to sound naive. There has been a huge amount of research effort put into that, obviously, and it remained a really stubborn problem. But I do think if you had unlimited resources and staff and maybe taking a different evolutionary and comparative approach could provide some insights. And I think in terms of neuroscience, we're living in a really exciting time because the kinds of tools to address questions that were previously practically impossible, that range of tools has really blossomed. And so I think I have all kinds of experiments I would love to do using organoids or assembloids, which are organoids of multiple cell types, using different primate species, and then doing gene editing to be able to mechanistically say, these are the genetic differences that are contributing to these differences in the human brain. And so people have actually been doing that a bit with, this isn't my field, but something I'm fascinated by, which is the evolution of development in the human brain, and have started to understand what are these tiny genetic differences, but that influence gene expression so that during development, they produce these really vast anatomical and biological, physiological differences between our species and other species. So I think there are emerging methods to try to do that in other parts of the life course. So that's the kind of thing I would do.
Definitely. And I think you're absolutely right that we're in this period in neuroscience, but I think in a lot of other fields as well, where the pace of advances is starting to accelerate as we get these new tools, as we're able to handle bigger, more complex data sets, involve people from multiple different fields to get a bigger picture of these problems than the narrow, very siloed approach that people have used historically.
Absolutely.
Well, thank you for leaving us something big to ponder for the rest of our day here, Elaine. And it's been so wonderful to hear more about you and your career, but we love to end our conversations by talking about advice. So do you have a piece of advice that somebody shared with you at some point that really helped you and you can pass on to listeners today?
I think the single best piece of advice I've gotten, because I've gotten so much good advice, is to maintain an interest outside of science. And I realize that not everyone listening to this is necessarily a scientist or a student, but I think that holds for any career. It's definitely a marathon, not a sprint. When you're doing research, there aren't quick results. And for that reason, it's really important to have not all of your identity tied up in science. I think something that happened to me, and I see a lot, is that when people transition into graduate school, they get kind of floored by the research process because it's very intensive and it can feel overwhelming. And so what people think they need to do is just double down and dedicate themselves more than ever to that thing. But because it's so challenging and intensive, that's exactly why you do need to reserve a bit of your energy for other things, because you don't want all your emotional eggs to be in one basket. I think inherently what you end up spending a lot of time and thought on, you invest emotionally in. And I've just found a lot of comfort in the things I do outside of science when my science isn't going well. And then you take a break and you come back and then it's fine. And to branch off from this, diversifying your projects is also helpful. That's advice I've gotten, and I think that's really good. Usually when one thing is kind of stuck, like this flow cytometry stuff, I still had another project going on that was going better. That's hard to do, for example, when you're doing your dissertation. I actually think that's one of the reasons that doing a PhD is so hard, is because you've got this one main thing and something always goes wrong.
Right. If it doesn't work, yeah.
Yeah. And it's on a fixed time scale, which is not how science should be because of all the reasons we've talked about. And so it's inherently really stressful.
Mm-hmm.
And I don't think I could do it again. But I'm glad I did now that it's over. And I think also when people say that maintaining an interest, they'll say like a sport or an instrument. And those things are really great. I wish I played to play an instrument. I love music, but I didn't. So even just making sure that I stay in touch with my friends who aren't scientists and talk to them regularly and things like that can serve that same purpose. Not to sound utilitarian about my relationships, because these are also people I love. But I think for a lot of people in research, it can strain those relationships and the other part of yourself that is not the researcher. But it'll make you a better researcher, too, to be more balanced, I really think.
Oh, absolutely. And I think when you're in these down periods struggling with your science, you maybe don't have the motivation to pick up a new hobby. Like, I should learn how to crochet.
Yeah.
I think just like you said, leaning into these parts of your life that you already have, but perhaps have been neglecting, I think is a good way to kind of reconnect, re-energize, and be ready to tackle those tough problems.
Yeah. And give yourself a reality check too. Most of these things, they feel so important. And it's not that they aren't. They are, but also there's just so much else going on outside the four walls of your lab or your office.
Absolutely. Well, wonderful message for our listeners. Elaine, do you have any other last note that you'd like to leave them with today? A last piece of advice or a last note of inspiration?
I'll just reiterate that don't be discouraged, especially if you feel like you've missed opportunities. It's not too late to try for what you're really interested in, and it can be really hard, and uncertainty doesn't make it easier, and that's certainly tough right now. I guess another aphorism is the best time to have started slang was yesterday. The next best time is tomorrow. And so that's actually also something I say to myself.
Yeah.
So do the thing.
I like that. Well, Elaine, wonderful note to end on. And if our listeners want to learn more about you and the work that you're doing, what is the best way for them to get more information or to get in touch?
I don't really do social media, but I do have Google Scholar, and I recently reactivated a website. So I do have a website, which is just my name, elaineguevara.weebly.com. Those are probably the best ways.
Perfect. Well, listeners, definitely take some time to check out Elaine's website. And Elaine, it has been such a pleasure to have you on the show today. Thank you so much for sharing your time and your perspectives.
Yeah, it was a lot of fun talking to you, and thank you for this opportunity, and thanks to everyone out there listening.
Well, it's been great to hear more about you and your work, listeners. Always fantastic to have you here with us as well. And we hope you join us again next time for another episode of People Behind the Science.